There we go. A quick welcome to country. So, just like to acknowledge the traditional owners of the lands on which we meet today. I'm in Melbourne, so it's the land of the people of the Kulin Nation. So, I'd like to pay my respects to elders past and present. Today, I'm just going to give a very quick introduction, if you were around a little earlier. Not sort of giving the what is Globus spiel because hopefully everyone knows what Globus is and why you're here. Just a very short update, and then we're lucky to have John Pearson from QIMR Berghofer give us an introduction to the SeqHaven project. And then we'll throw it open to your questions, see how we go. This is something I would have presented, I think, at the last office hours we did. Was pretty much our work plan for this year. Everything is humming along. You'll see the link up there in red on the right-hand side. We've been able to, with all the support of all our Globus subscribers, put a registry together. So that web page sits in the aarnet knowledge base, lists, I suppose, contact details for all the Globus nodes across the universities. So this is at face value, it's a web page, but I think it's extremely valuable. One of the consistent bits of feedback we get tend to be from researchers asking, does my institution have Globus? Or if I need to repatriate my research data, how do I get that home? How do I move it? So this is going to be a fantastic thing for people to be able to find out who to contact and what Globus services are available to them. The other thing I'll just give a shout out about is the dashboard. We've been working closely with all the Globus subscribers to put together a dashboard to, I suppose, give us a little bit of visibility in terms of usage of the service. And we think that's going to be really valuable. Primarily for our reporting to management, so we can indicate the services is being utilized. But as you can see from those use cases too, it actually will give us some data to be able to have a look to see, what's actually going on, what are the main data channels, are there things that we can do from a network perspective to help bolster that. So that's it for me. I'm going to hand over very quickly now to John, and yeah, we'll go from there.
So I'll introduce myself first. My name's John Pearson. I'm a cancer researcher from QIMR Berghofer, as Greg said. I wear a second hat. I'm actually also responsible for the high performance computing platform here at QIMR Berghofer. And this project that I'm going to talk to you about today, SeqHaven, sort of sits, or the Globus project. So it sits at the confluence of those two hats. It's sharing genomic data using IT infrastructure with Globus as the software. So that's what I'll chat about. So I'll give you a very quick primer on on just what a genome is, so you know what we're sharing. Every cell in our body has a copy of our genome. Our genome is something we inherit from our parents, half from mum, half from dad. The chemical that makes up the human genome is DNA. So you'll often hear genome and DNA used interchangeably, and that's why. For the purpose of doing analysis, there are four subchemicals in DNA, and we have a letter for each of them, A, C, G, and T, which means by and large, genomic analysis is actually a string operation. Now, there's 3 billion letters, and in fact, we have two copies of the genome in every cell because we get one copy from mum and one copy from dad. The sperm and the egg each only have one copy, but every other cell has two, and that's why we're a mix of mum and dad, because we have a full copy from mum and a full copy from dad, which means we're all carrying the two copies, but only parts of each are used. So 3 billion letters, and I'm primarily a cancer researcher, although we do a few other things. And what we're looking for in cancer is a couple of those chemicals change. That doesn't sound like such a difficult problem, but it unfortunately it is, because one of the hallmarks of cancer is unrestrained mutation. That's how the cancer escapes from being killed. Your body is killing cancer. Each of you sitting here at the moment It's weird to say, but we all have cancer in us right now, and 999,999 times out of a million, our body defeats it. A cancer is just a badly behaving cell. That happens all the time. Our body almost always kills it. Once in a while, it doesn't. But by the time a cancer makes us sick, the few changes that started the cancer, they're very difficult to tease out under the thousands or tens of thousands or hundreds of thousands or millions of additional mutations that cancer has layered over the top. So it's this weird forensic effort of trying to work out what must have happened first. And the pattern is different in different people. If you take two breast cancers, they won't have the same mutations because we have many genes, and there's many ways or combinations that can go wrong that all lead to cancer, because cancer is really just a process of a badly behaved cell, and there's many ways to get to badly behaved cell. And if cancer was easy, given the number of Nobel Prize winners who've spent their entire careers chasing it, if cancer was easy, the tens of thousands of us working on it, we would have fixed it. So cancer is tough. Okay, just a quick thought exercise just to get the scale of the data. So if you had marbles, a different color marble for each of the four bases. For the Australians on the call, this is the Melbourne Cricket Ground, one of the great cricket grounds on earth. It's about 20,000 square meters, so about four standard football fields. If you went to the middle of that and started laying down the marbles, one marble for each of the 3 billion bases in the human genome, how much you each have, this is a thought exercise. I'll give you a moment to think about how much of the MCG do you think we would cover in marbles to represent the 3 billion bases. Okay, the answer is we need 15. We need 15 MCGs to hold one copy. So 60 football fields, and because the sequencing machines that we use to investigate the genome, they make errors about 1 in 500 or 1 in 1,000. So in fact, we normally need to do 30 copies of the genome approximately in order to be confident that we'll see past any errors that the machine's made. The machine errors are mostly random, so that's a little sprinkling of error everywhere. And also for every one of those observations, we get a quality score, which is saying how likely we think the observation is real. So for one person's cancer genome, we usually end up with about 100 billion observations, each of which has a base and a quality score. And that's what we dive into. Okay, so what do I have to share? So one human genome compressed data is about 100 gigabytes. In cancer, we need to do 3 genomes worth of sequencing. Because everyone's a little different, it's actually no good comparing. If I sequence a cancer, it's no good comparing to some nominal human reference because we're all about 3 million bases difference from each other. That's why we're all a little different. We're basically all human. We're 999 out of 1,000 bases the same, but with a little sprinkling of difference. So when we do cancer, we need to sequence the person themselves, so usually their blood, and their cancer, so we can see what changed in the cancer. And because surgeons, it's impossible to just hand me a sample of cancer. There's always a bit of not cancer in there. We have to
get that.
we have to sequence the cancer twice a day. So three genomes effectively for one cancer patient. So that's 300 gigabytes. A small study these days is 100 patients, so 30 terabytes is a small study. And there's a much better sequencing technology that's just come out, but it's about 15 times as much information that we have to process. You have to process about 30 million observations a second on a voltage sensor. So those voltage across 72 hours and 12,000 pores, each with 5,000. I think it's 5,000 observations a second per pore. The data gets big. Okay, so we need to share it. So here is the SeqHaven project. Okay, so this is what we had to put together. So we've been wanting to do this for about 6 years. It's been very hard to find someone who wanted to fund the infrastructure. It's easy to get, well, it's not easy. it's easier to get funding to do a bit of cancer research than it is to say, I just want to buy a heap of computers and share data. But we finally found the folks at NCRIS and Biocommons agreed to fund us. So this is what we built. We built, in order to make my organization less terrified of what I was going to do, we built a whole second network. So we don't have any real connection to QIMR's main network. We have a separate network, security appliances. We went with Proxmox, so we have 12 Proxmox servers, each attached to a disk array. So we're running Ceph, and none of that worried me because hardware is pretty much a thing that my team does in and out every day. And we honestly just put the software aside. Failure of communication on my part, but in the back of my head, I was so concerned about the hardware. In terms of sharing the data, all I really had in the back of my head was it would be something like FTP. And having written software for 40 years, I just had in the back of my head that if I didn't find a piece of software I liked, I'd just make one. So I wasn't even thinking about the software until we got the hardware in place. And then we arrive at Globus. Okay, so this is this may not be interesting, but I thought I'd at least tell you how we arrived at Globus, given that we are very new to Globus. We're up and running, but we're probably only really 6 months into this process. So what brought us to Globus firstly was, as academics, knowing that Globus was made by the University of Chicago. So it was folks who think like us making software for us. That was really comforting. So folks who, at least some of whom are even probably in our domain of genomics. So that was a huge plus, knowing folks like us were making software for us. Longevity, I've been a part of and adopted so many dead projects across the 40 years I've been doing this cancer research. The fact that this has been around for coming on 30 years, huge plus. Lots of use already, 1,500 organizations. Lots of local use by friends and family effectively, which I hadn't even really realized. So the two big universities in my hometown here, University of Queensland and Queensland University of Technology, they're both Globus users. NCI and Pawsey are the two big federally funded research supercomputers in Australia, both Globus users. CSIRO is our national science. It's like the NSF, I guess. It's a national science foundation, or national science organisation funded by the federal government. They're users, and then many of our collaborators, University of West Australia and the Kids Research Institute, are just two examples. So wildly commonly used, which I hadn't even realised. Local support. I can talk to Greg. That's incredibly useful. Web command line API. This means no matter what level of sophistication, I'm relatively confident that the folks I want to share data with are going to find something, a level that they're comfortable with, and no matter whether they're relatively naive or relatively sophisticated, the system will cope. Clients don't need a license, and we've got the freemium model for server, and of course we're paying for support because we want some of the extra goodies that we haven't used yet. That was why Globus. So where are we up to? We have done 3 pilot transfers, 2 international for us and 1 with a national collaborator. And what we've learned, so these are all things I didn't expect and hadn't really factored into the way I was picturing using SeqHaven. But the first thing was 2 of the 3 already had Globus. This was really this has proved to be incredibly valuable because it means that the conversation about transferring the data is sysadmin to sysadmin. So data is moving to those 2 organizations just like water through a garden hose. That it is just utterly free. We don't even have to worry about the user at the far end, whether or not they can actually get the data, because they'll work with their sysadmins at their end to get the data. So that's incredibly freeing, and it made those data transfers a lot simpler for us in that we weren't providing any support for our consumer at the far end. They were integrating, or they were interfacing with their own sysadmins at their organization. So unexpected and a big deal. There was obviously some learning, and we're still very much in the early stages of this. There was a significant amount of negotiation, shall I say, with our IT organization, trying to get firewalls, etc., configured so that we could get the things we needed to get done. But I think that is all sorted now. And this was the big one. But again, this is failure of imagination on my part. But I really, having adopted Globus, I'm really thinking about a lot of things that we would never have thought about in the past. So in the past, we would send our data to Europe, actually, to EGA, because we didn't have the capability to share. And there's two huge genomic data repositories on Earth, one in the US and one in Europe. And the vast majority of researchers send their data to be shared by one of those two organizations. So that means, in my head at least, data distribution is very much a case of sending it somewhere and then someone shares it for us. So it's a pure distribution play. But we have discovered, now that we're using Globus and sharing ourselves, it's an incredibly powerful collaboration tool. One of those three folks that we did the data transfer to, we ended up in discussions with them. It turned out they couldn't take all the data because they were a smaller organization, and they wanted an 18 terabyte data set from us, and they didn't have the capability to hold that themselves. They wanted in little pieces. So we said, well, we can actually send you any data you want. You don't just have to have the raw data we originally talked about. So they then used Globus to send us some examples of the way they like to see their data. We used Globus. We did some work at our end, sent them data via Globus. They have a Globus install. They took a look at that data. We went backwards and forwards. We've probably done at least 6 shuttles of data backwards and forwards with that user, all essentially from the point of view of the researcher. Globus has meant this is an utterly abstracted transaction for me. I talk to my researchers at the far end, and then in the background, our two teams of sysadmins and Globus admins just move data backwards and forwards incredibly easily. So being out of the loop as a researcher actually it's really empowering in this case, and that's because the two groups of sysadmins are just shuttling the data via Globus. So that's the first thing. So we've got what would have been 3 data distributions has turned into 2 data distributions and a collaboration, and that's huge value for us, and it's also huge value for cancer, because it means that we're going to make a lot more use of that data than the two groups would have separately. Us sending data to someone in the USA. Instead, now there's an Australia-USA collaboration going on, and we're going to deliver much more value. So good for cancer as well as good for us. New data set types. In the past, we only ever shared raw data. That's what everybody does. We just send raw data. and that's all any of the systems are really set up for. But having control of the means of distribution under Globus, we have proposed to our funders, and they're very excited about it. We're going to share at as many levels in the value add chain as we can. So for labs that are big and powerful and can cope with the raw data, with downloading tens or hundreds of terabytes of data, they can have the raw data. But we're going to also start sharing analysis sets. So we already do the work, all the analysis work for our own internal research. We're going to start sharing some of those data sets. So if you can't cope with the raw data, but you'd still like to get lists of the variants of the insertions or deletions or substitutions, all the different types of variants, we're going to share those as data sets. So we think that will open up the data to a lot of smaller labs who would have possibly looked at this data in the past and said, I don't have the resources. I would love to use some of those big data sets, but I can't cope with the resources. Or I can't come up with the resources to do the analysis for myself. But because we control the means of distribution, we're going to start sharing these new data sets and try and get additional value out of the data. The underlying the rationale, if you like, for the sharing is we have derived so much value from the data. my collaborator Nick Bordell and I, we have 10 Nature papers that we've generated from this data. So we've been incredibly successful with the data. But even with all of that, we still only ask a limited number of questions. We've got 20 people in our combined teams. There's still a limit to how much 20 people can do. So the intent with sharing the data is we would get much more value out of the data if there's 1,000 people using the data. So the more people who use the data, the better it is for cancer. So sharing is sort of it's a fundamental mindset. If you don't share, you're doing it wrong. You've got to share your data. And this is a thing that we had never considered before, but we're now thinking about user contributed data sets. So if folks are big enough to take our data and do an analysis, we're going to ask folks when they download the data if they would consider, if they do something clever with the data, would they consider handing us that clever data set and let us share that as well? And this is just sort of trying to add even more value on top of the data. The data costs us millions of dollars to generate. So every time someone does an analysis on top, that saves someone else doing millions of dollars worth of sequencing. If we can just keep adding value on top of the work, the sunk cost of the sequencing we've already done. Okay, this is the future. So it's already radically changed the way I think about sharing data from just being simple raw data to being there's really no limit on my ambition in terms of sharing. I don't even know all the things I can do yet, but I know I can do more than I'd ever thought. Things that we're not doing, but that we'd love to start getting into is Globus flows. We already have we use workflow definition language (WDL) or for all our workflows, but we'll have to have a look at Globus now. We're using it. We use NiFi for internal event-driven work, Apache project, and we're still old-fashioned cron folks for scheduling. But Globus has all these facilities built in, so I can imagine there's a chance we'll become a hybrid shop where some things where it makes sense for Globus to do the work for us, we'll do that through Globus. Other things we'll continue doing it the way we used to do. And all the connectors, we haven't even taken any of those out of the box, effectively. Those are all just things for the future. That's it, gang. So we're very early. We're very excited, and really, it has having control of the means of distribution and having a system like Globus has just completely changed the way I think about data sharing. Thanks, folks.